In this video, we'll be continuing on talking about hemostasis. Hemostasis, and the part we'll talk about is coagulation. Now, coagulation is like the gold standard of stopping the blood flow. And this is really the process of making a blood clot. So coagulation is the last and the most effective defense against bleeding. And it's really based upon having pre-made inactive factors that then become activated. Now, why is it so important to have these pre-made factors? Let's say you cut yourself. You certainly don't want to spend your time turning on genes, making proteins, getting the proteins to the right space or location, and then having a clot. That would take way too long. So the game plan is to make these pre-existing factors, which are not fully activated, and then activate them in what we're going to see is called a cascade. And what we're really looking at is the conversion of a plasma protein called fibrinogen. And that, the goal is to turn it into fibrin threads, which will form basically a mesh. These threads will form a mesh, which becomes almost like a skeleton for the blood clot. Now, these procoagulants, these clotting factors, typically are usually made in the liver and are present in the plasma. As I said, what happens is one factor is activated, it then activates the next factor in the step, in the pathway, and then etc., etc. And this becomes part of what we call a reaction cascade. Now, there are two ways to initiate this, and the two are the extrinsic pathway and the intrinsic. And of course, the name gives you quite a big hint, is that the extrinsic is where we have factors released by damaged tissues. So it's the tissues. Whereas intrinsic, the factors are found in the blood, intrinsic in the blood, and begin the cascade. And of course, this is really tied in with platelet degranulation. This is sort of the gold standard that we want. You can see the fibrin threads. You can see how that mesh forms a backbone. How we get trapping of red blood cells, platelets, etc. And this is really becomes, as I said, almost a skeletal system for the forming blood clot. Now, here's the basics of the pathway, and we're going to quickly walk through it. As I said, you have the extrinsic and the intrinsic pathway. Now, let's start with before we actually go there, let's have a look. Here's the intrinsic. There's the extrinsic. You notice each pathway leads to the same point, and that is turning on factor 10. Inactive factor 10 is activated. So let's start with the extrinsic. The extrinsic, it's damaged perivascular tissues. Basically activate factor 3 in association with factor 7. Calcium plays a role, and that will lead to the formation of active factor 10. For the extrinsic, there's a different pathway. The platelets lead to the activation of factor 7, factor 11, factor 9, and then factor 8. So you can see how this one leads to that one being activating that one. And it's not just one makes one molecule. This factor can actually do it to a number of inactive ones. This one to a number. So we're going to end up with a very extreme growth and increase in number of activated factors. Now, once we get down here, and this is where we're really going to spend most of our time and energy, we should know that there's an extrinsic and intrinsic. Both of these lead to factor 10. And that's where we're really going to pick up our knowledge base here and really focus in on here as a class. Is that factor 10 so inactive factor 10 now becomes activated. There are some other factors, 3, 5, calcium, and PF3. Okay, we're not going to talk much about PF3 and the role of calcium right now. But factor 10 then becomes or initiates the prothrombin activator. Now look at the name, prothrombin. So it tells you what it's going to activate. It's turning on prothrombin. So we take prothrombin, and if you're called prothrombin, that sort of insinuates you're before thrombin. So not surprisingly, prothrombin activator takes prothrombin and turns it into thrombin. Now look what happens. The thrombin itself has a role down here, but let's not ignore what's happening here, is that there's a feedback mechanism. And you can see this becomes a positive feedback, which of course you'd expect if you want something to happen in a big way and fast. So the thrombin takes fibrinogen and it turns it into fibrin. And the fibrin, in association with another factor in calcium, becomes the fibrin polymer. And what do we mean by that? That's that mesh of fibers that we looked at before. Just that big mesh where we have, red blood cells and platelets stuck within it and other materials. So we're just going to go over here to the side and just remind ourselves you can start from the extrinsic pathway or the intrinsic pathway. And in both, calcium plays a role. Now, I just want to talk briefly about the enzyme amplification. That's the concept that where, if I activate a little bit of the first factor, that can then, it only doesn't get used once. It can actually activate a number of the next factor in a row. And each one of these can basically activate a number of its target. So you can see what happens is very quickly, by having multiple steps, I can start off with very little of a factor, and by going step to step to step, I get an amplification, and by the time I get down to the end, I end up with a whole lot of product to put this clot together very quick. So by having a multi-step process, it allows for the enzyme amplification. Also, if you want to think about it, it does give you some opportunity for maybe some more control points whenever you have a system like this. And also, as we saw, there's a built-in feedback mechanism that is part of this. And just to sort of quickly recap is that, we have activation of factor 10, which leads to the production of prothrombin activator. Prothrombin activator converts prothrombin into thrombin. Thrombin itself converts fibrinogen into fibrin, and the fibrin becomes part of the backbone of the clot. And as I mentioned, we also have a positive feedback loop that is critical to this to make sure it happens in a big way in a short period of time. Now we're going to leave it there for now.